Hello, my name is Jillian and I am a bioinformatics infrastructure consultant. Today we will be talking about three things. First, we will do a brief introduction to CellProfiler on the command line. Then we will be talking about how you can batch your analyses using CellProfiler. And finally, how you can actually profile your analyses, profile your CellProfiler analyses, which is to say how much memory and CPU they are taking so that you can make informed decisions about how you want to batch your analyses, how you want to split them, how you need to group them, all that kind of thing when it actually comes time to run them. So with that said, let's get started. This is one of the images. Isn't it nice? I like cell images. I think they're very pretty. So anyways, we are going to be using two data sets today. We are going to be using this human cells data set from cellprofiler.org. And we are going to be using this 0 to 1 human cells for, I think they're cancer cells, and they're testing the effects of different chemicals on the cancer cells. I'm not a biologist, so I'm just going to get that out of the way, but I thought that I would at least briefly say what are the data sets that we're using. So what we're going to go over are some sort of basic instructions. Don't worry, all of these will be listed below. There will at some point be a blog post. At some point. So what we're going to do is we're actually going to run CellProfiler using Docker, and we're going to do that because one, I really like Docker, and two, it will allow us to much more easily profile the instance where we need to find out the memory and the CPU. So with that, let's get started. I already ran this, so I'm not going to do this, but what we have here is we have a Docker file, and we're using the base CellProfiler image. Now, the base CellProfiler image is really great for probably most of what you're going to want to do, especially when you're in a job queue environment. But for this particular example, what I really wanted to do was to be able to just treat the Docker container like a computer and just get into the shell and just kind of play around and really do a more exploratory type analysis. So that's why you see this. Also, tree is really nice for investigating file structures, wget for downloading the data, and ImageMagick for some post-processing that we're going to do later. So with that, let's get started. If you need to build the image, this is the command to do that. I have already done that, so I'm just going to run this. Okay, and then what I am doing is I am saying that I want to have my current working directory, and I want to make this available in the container as /project. So if you haven't ever used Docker before, it's pretty much, it's a kind of like a virtual machine, but you can treat it like a computer. So that tends to be what I do most often, at least for exploratory analyses, is I will just do this. I will make the files that I need available in the container, and then I will just drop into a shell, and then that's it. It's a terminal. It's my computer. So what we're going to do here is we're going to go to the example data set. And you can see it's all here. If you don't have it, what you do is you just run this command, wget, unzip, and then this is the file structure that you should have. This is why it's nice to have tree. Like, let me show you tree. Oops. So what happened is that I already did this. And then, I don't know, my screen recording program just freaked out and didn't keep anything. So that was that down the drain. So you'll see there is some output, but we're going to remove it because we're going to do this fresh. And then let's run tree again. Okay, so you can see we have our example human.cp pipe, which is the CellProfiler pipeline. We have a README. There wasn't much in the README. It just says where the data, no, it's a different README. It's complicated having a million READMEs. Yep, so these are images of human cells were provided by the JSON mitotic index experiment. I'm assuming that you guys know what that is. And we have some images. So just to be clear, for this data set, this is not a particularly interesting data set for batching data because there is only this, and you could just run it and you could be done and not spend time thinking about how you're going to batch. But this is the introduction to just using CellProfiler on the command line. If you're used to using CellProfiler, you've usually opened it up in like a GUI kind of platform. When you're running like an analysis pipeline, usually you're just running that just strictly on the command line, particularly when you get into larger data sets and really like high scale experiments. So what we're going to do is we're just going to run this. There we go. Okay, so I'm saying CellProfiler --run headless. This means don't try to open up the GUI because we're in a Docker container and we're not doing that. And then I'm using this pipeline. This is the output that I'm deciding. For this example, this can be anything. Depending on your analysis, you might need to structure this in a particular way. We're going to talk more about this when we get to the next example. And then my input is images, and you can see, here it is. Here it is, we're all good. We're here. And then you can see right here, okay, so we have an overlay, and then we have our cells. Hopefully you all know what this means. But anyways, we have data, and presumably it's very nice data, and we would want to do something with it afterwards. So that's the end of that example. Now we're going to move on to this more interesting example. Let me get rid of some of these files because they take up my screen real space here. Okay, so if you need to grab the data, this is what you can do. We're only going to be using this first data set, this week 1_22123 for today, but if you really wanted to go through the whole process, you could do this for everything, right? And then you also need to go through and you need to get some of these data files, the analysis and the illumination pipeline files as well. And then, you know, in order to kind of understand, let's go take a quick look at this. So if you wanted to understand the metadata, you could go, you wanted to understand the actual experiment itself, not what I'm talking about. I'm mostly just talking about batching it. You could actually go here to the website, and then they also have a paper published, and they have everything here. So everything that I've downloaded, where is it? Here? It's all coming directly from this website. Here, these are all the data sets. And these are all the data files, so it's nothing. It's nothing like too scary or too out there, right? And then these are the analysis pipelines, and that's it. So I do kind of want to say I had to figure out a bit exactly how to run these, but it's not too bad. Generally, you do illumination correction first, and then you do your analysis. So that's the way that we are going to run this. So you will actually see, let's go back to the site here for a minute. This is a very large data set. So you have each one of these archives is 750 megabytes each. I'm not sure how much it is unzipped, but you got to figure probably around a gig per data set, and you have multiple weeks, and each one of these is split. So this is a perfect example for batching. You would not want to run this all serially. If you did that, that would take forever. It would take the rest of your life. Your paper would never get out. It would be bad for everybody involved, right? So this is what you want to do. This is why you would start to batch your analyses. And what batching usually is usually you're running things in parallel. So let's say you have an analysis, and that analysis has 4 steps. Each one of those 4 steps takes 15 minutes. That would be an hour if you ran those, step 1, step 2, step 3, step 4. But what you can start to do is you can start to think about, okay, how can I parallelize my analyses? How can I take these steps and separate them so that I'm doing everything in a parallel fashion, which means, say, step 2 doesn't depend upon step 1. You can run these in parallel. You can take each one and you can run it as sort of its own separate analysis. You can think of it that way. So if a step in analysis can stand on its own, or multiple steps in an analysis, they can stand on their own. Usually you can run those in parallel, and that's the idea that we are getting at here with the batching. So what we would do is we would say run this and run this and run this and run this. So say if each one of these took 15 minutes, we could run them sequentially. That would take an hour. If we run them in parallel and all 4 were running at the same time, it would only take 15 minutes. That's the idea there. That's what we're trying to accomplish. So now that we have that, one of the things that is provided here is this data file. I think that's what CellProfiler calls it. Yeah. So is this one? So this is kind of a description of a mapping of the file names to their experimental conditions to their replicates. We're not going to be using this to batch our analysis specifically, because you can do this in CellProfiler just by giving the first and last flag. But it is a really good idea to look at this, because if you look at this, it will give you an understanding of how the data sets are structured. And if you ever have to go through and batch an analysis, You need to understand how the data sets are structured. I don't know why this keeps happening, that I keep jumping to the beginning of this file, but that's okay. Okay, so first what we're gonna do is we're gonna run this illumination pipeline. Actually, I'm doing something a little bit silly. So for the illumination pipeline, this is one of these things we can't actually batch this because what it's doing is it is generating an average across all the images. And for that, it needs to have all the images in the data set. So I am giving these first and last flags. That's just for the purposes of this tutorial so that it will get finished quickly. Otherwise, it takes quite a while. I don't remember. I ran the whole thing. I don't remember exactly how long it takes, but it takes some time. So what you want to do is you want to run through this, and then you will see, oh, look at this. Yay, CellProfiler is running. And then you will see it's coming up here, and it will tell you which image it's working on, which is pretty nice. It will say image number 1, and then, you know, it will go through images, metadata, groups, correct the illumination, and it's calculating. And it will tell you the amount of time that everything is taking. I was just about to do the Portainer thing, but that's fine. Okay, and then we can go and we can look here. And we can see, this isn't something that I can actually open because they're like NumPy. Oh, they are NumPy files. So you'll see here that we have this illumination actin average. So this is why you would need to run these. Like don't do what I did. Don't use this first and last flag. Make sure that if you're actually running this pipeline that you're running it without this because it's doing that. And then also, normally I like to keep my inputs and outputs separate, but for this one, the next step in the pipeline, the analysis step, actually depends upon the output from this pipeline. And in this example, they were all kind of structured in such a way that it expected to see everything in the same directory. I had to kind of like play around with that a little bit to figure it out. You might have to too. That's why it's really nice to do things like this where you're just doing like just absolutely the minimal example. So now that we have that, let's go through and let's run. Oh no. So this is how you would run the illumination pipeline here if you wanted to run it for the entire data set and go through and do this properly. I'm not doing that. Okay, and then what we want to do is we want to run this. All right, so now what I'm doing is I am saying run the analysis. This is the input. The input is our week 1 data, week 1 underscore 22123. And the output is this week 1 underscore 22123. And then I'm also giving an output per batch. And that is because you don't want for CellProfiler to overwrite files and to clobber each other. I think whether or not it does that probably depends on the analysis steps that you're taking, but just to be safe, this is how I'm doing things. So there we go. All right, so now that this is going, what we're gonna do is we are actually gonna see how much memory and CPU our images are taking. What is this Portainer stuff? Oh, here it is. Okay, so I already ran this, so I'm not going to run this again. Yeah, see, look, it says it's already being used. That's fine because it's already running. But this is a really simple way of doing memory profiling on your applications. There are, of course, always many ways to do absolutely everything when it comes to software. This is my favorite way just because it's really easy. You just run this and that's it. And then you get a nice web interface. If you're starting this one up for the first time, it will prompt you to create a username and a password. I've already done that, so I'm just going to go on over here. I am going to see my containers. Here is a running container for me. It's CellProfiler. Actually, when you do Docker run, you can supply a name. Otherwise, it just generates you a name. And then I'm going to go and I'm going to look at the stats. And you can see here, it actually says, okay, this is the command that's running. So we know which command is running. I'm going to tell it to update every 1 second because that will be a little bit more interesting for us. So you can see here it is. It's on 500 megabytes. It is actually going to jump up to at least 1 gig. I think it might jump up more. All right, so that's the trouble with doing that. Let's do this again. I don't know. Every once in a while, I don't know why it just freaks out like that. But it does. Here we are. Okay. So then kind of the downside of doing things like this, the way that I'm doing to get the memory in the CPU, is that you have to babysit it a bit. You have to watch in real time. There are plenty of other programs that can give you a nice output that will live on. But I mean, really, for just kind of really simple profiling, I like this way of doing things. This is fine for me. And you can see it will go and go and go. I wouldn't be surprised if there was a way to export the stats. I just don't know how to do it, because usually I just watch it in real time. Okay, and you can see it's going, and you can map kind of the times. And then you could get pretty into this, because CellProfiler actually gives you, also gives you these timestamps. So you could really get into it and see exactly where it's using the most memory and CPU. This will, at some point, this will kind of jump up to about a gig, maybe a little bit more than a gig. I'm not sure. But, yeah, but there we go. So the whole point of doing this is, say you had a machine and it had 32 gigs of RAM, right? And you know that each one of your CellProfiler analyses is going to take around 1.5 gigs. And then let's say you're like, okay, well, I want to make sure that I have a bit of a buffer, so I'm going to say 2 gigs. Then you could do 32 divided by 2, which is 16, right? And then, you would want to leave a couple gigs for system processes. Let's say maybe you do like 12 to 14 in parallel. So let's say that each one of these analyses would take an hour. So if you're doing 12 in parallel, instead of taking 12 hours, it's only going to take 1 hour. That's pretty significant, especially for a data set like this, where, how many of these do you even have? You have 55 zip archives. So when you're getting to really large experiments, this kind of time saving, it does get to be very significant. Also, depending on the analysis type, some programs will actually read all the data into memory and then analyze it. So then you can also have some really huge memory consumption requirements. So you can see, all right, this is still going. This is still going. Let's see, do we have any kind of interesting output? We might. Okay, so you can see we are actually already getting some output. These are the very dark cells. These are the very dark cell images. This illumination corrected. So you can see actually for the illumination corrected, it does, like you can see a little bit more. If I'm going to do human readable images, I actually use a program called Image Magic for that. Okay, so then you can see this is finished. It exited. Did it exit successfully? Yep, it exited successfully. So if you need to know if something exited successfully or not, the exit code for something that ran and there was no problem is 0, and you get it from this command echo $?? So let's go up a little bit here. And then you can see this very, very, very last step here is export to spreadsheet. So let's kind of go take a look at that. All right, so the very last thing that we have here is this export to spreadsheet. So you can actually see that this would have been overwritten. This would have been overwritten if I'd used the same output file per batch, and that's why it's important to make sure that you use a different output directory, not file. They use a different output directory per batch. So you can actually see this is, I think this is a very verbose, this is kind of a very verbose version of our initial analysis input file that gets output for I would imagine for record keeping. Really, I'm not really too sure. So then what you could do here, so anyway, so this whole measurements file here, this one would get kind of clobbered, but then you can see here that we have this, the cells, and then we have, we have each one of our images here, and then you have a whole bunch of measurements that I don't fully understand. Oh, area. I know what area is. Look at me. I know what some of the biology is. I know what area is. And then you have a whole lot of other things. I'm pretty sure these are the things that you supply through the CellProfiler analysis file, and then you get your numbers at the end. So you can see there's a lot of stuff that happens in here. Okay, so there we go. So there's that. Do we want to see any of these? Oh no, this is just some data about the image. And some data about the nuclei. All right. I will not play a biologist on the internet. That would not be good. But one thing that I did discuss was, I did say, one thing that's a little bit nice, if you want human readable images, these images, they will not be suitable for analyzing in CellProfiler. Just to be clear about that, this is if you want something human readable, you want something to put in a paper, you want something to put on a poster, you want to have like a nice desktop image that's a cell, this is what you could do. If I can get to it, where is it? Okay. So this is what we're gonna do here. I'm gonna go to these images here, and I am actually gonna convert these. I'm going to use ImageMagick. So the program is ImageMagick. It has a ton of image processing utilities, and I am going to say convert it, do these auto levels, keep the quality really high, and that you can go from TIFFs to PNGs. And then let's go find this. Okay, so then you can see this is the TIFF. It's really dark. I can't see anything. CellProfiler can totally make sense of this. I cannot. So if you wanted something that's human readable, then there you go. You could use ImageMagick and you could get something. Let's go look at that. See, this is nice. And then there's this one. And then there's this one. I think actually most of the time when you open these up, if you open them in CellProfiler or Fiji or I don't know, any of the other ones, they tend to look like this. But then if you just open them in the system viewer, they look like this. So, I don't know, mystery for the ages. But that's that. Let's go see if we can get anything cool from the Portainer. All right, so then you can see on the Portainer, actually this command is finished, but we didn't exit the Docker container, so it's still in here. And then you can see here is our memory usage. I really kind of thought it topped out at around 1 gig, but maybe not. And then as soon as the command finished, there we go, then it went down to nothing. So that is how you would go. You would take your CellProfiler, each one of your analyses, you could start to think about how you could batch it. What I like to do is I like to take like paper and pencil and actually draw out the steps in the analysis and then kind of figure out exactly where it is that I could start to split things. And then once I split them, okay, do these steps depend upon each other? Can I parallelize them? So that's it for that. Was there anything else I was going to talk about? I need to have a better plan for how I end these videos, because otherwise I just end up feeling very strange. That's it. I'll leave you with one of these nice images. There we go. So that's this nice image. This is how you would split and batch all of your CellProfiler analyses. I hope that was helpful. Down below will be a link to the blog post and a link to my website if you would like to learn more. And thank you. Have a nice day.